Hello. In continuation of generation of features which we completed in the last two classes. So actually 5 phases are there for the screening of phytochemicals. In the beginning, you collected data, then so it's actually a phytochemical data which you collected from your database or from any standard database, from which you identify a target data, probably those which are required for bringing down the platelet count, or those which are useful for a particular type of cancer, those which are useful for inflammation, those which are useful for preventing the viral infection, or whatever may be that. So you have to select from the data, from the raw data you have to select, and that process is what is known as selection. And so you will be getting a target data after the selection process, or what you call as after the first process of data mining, right from a raw data to a target data you have come up. Now we have to do some pre-processing, like some of the molecules may be having very small molecular mass, some of the molecules may not be suitable for drug action. You may think like, okay, number of hydrogen bonding or hydrogen bonding acceptor, some common features which you are using, you will be able to apply, and based upon that, sometimes we'll be able to make some pre-processing. And so actually from the target data we have come to a pre-processed data in this phase. Then from the pre-processed, again more features are going to be added, and by introducing pharmacophore features or any other features, we'll be getting the next phase. Refined data we'll be getting, a transformed data we'll be getting. So this is what is known as transformation. Each phase, it can be in one phase or it can be in number of phases. So sometimes I may tell you, okay, you add one more component and see whether the data has been transformed from a pre-processed level to a transformed form. And once you get the transformed form, that you have to very closely observe. The transformed data you have to very clearly observe to see the patterns, identify the patterns, understand that data and get the pattern, and that phase is what is known as data mining, right? And once the data mining step is over, now you will come to what you call as the pattern to knowledge level. It's because already pattern is with you. That pattern you closely analyze, closely study, then get some knowledge, get some information, and with that you interpret the data, you interpret the results. So these are the normal phases of data mining that is known to you, isn't it so? And so far we have seen some primary features like molecular weight, and we know that, see, a very big molecule means that would not be suitable as a drug candidate. It would not be a lead molecule, it won't be a potential molecule most probably, because the compounds with higher molecular weight, they are less likely to be absorbed, isn't it so? So if it is not going to be absorbed, you know that the absorption, distribution, the pharmacokinetic and pharmacodynamic features, Absorption comes distribution, metabolism, excretion, atmy, and finally toxicity. So these are very important phases which a drug molecule has to pass through. So automatically, if the molecule is not going to be absorbed properly, then no question of keeping that molecule, even as a trial. And normally, you see, scientists or people who are working in this area, their objective would be to design and develop a drug molecule with minimum molecular mass. Molecular mass should be as low as possible. So that process would be minimization of molecular mass. That optimization, because most of the organic molecules will be very massive molecules. So during the synthesis also, you have to see, suppose you want to change a template into a drug molecule. So that modification, within that modification also, make sure that that molecule is not going to be a big molecule. So our motive will be to bring down molecular mass as low as possible. It's seen that more than 80% of all identified drugs so far, reported drugs so far, traded drugs so far, they have a molecular weight less than 450. So our objective while designing drug likeness or while identifying drug likeness in a phytochemical, see that that molecule is not very small. If that molecule is going to be very small, it may be turned up as a pseudo drug or decoy. If that molecule is going to be very big, then it may not be absorbed. So that is why that molecular mass, it should be a primary feature. And we have made some, condition or some sort of value to molecular weight. And the second factor which we have used earlier in the previous classes, that is log P, right? And you know that log P is a very important property as far as drug molecules are concerned. So actually, normally what you find is log of partition coefficients between normal octanol and water. Means what you can see is log of concentration of drug molecule in octanol divided by concentration of drug molecule in water. It's a very simple experimental value, in vitro measure, and log P helps you a lot basically. And we know that as log P increases, chance for absorption that decreases. So a higher log P normally results in a low bioavailability, right? And compounds should have a probability, it should have a higher probability to be absorbed because it has to pass through the cell walls, cell membrane. So membrane permeability is going to be a major factor. This needs to looked into. So into that, say as log P increases, we know that it's a measure of quantity of that substance in octanol divided by quantity of that material in water. So as log P increases, chance for that molecule to be distributed in octanol increases, right? So its lipophilicity increases, hydrophilicity decreases. And so automatically we know that for any molecule to pass through cell membrane, it should not be very hydrophilic, it should not be very lipophilic also. So it is seen that log P value must not be greater than 5. that is already known to you even by the very preliminary Lipinski's rule. Same thing has been informed. And if you take the log p value, there also a same result would be obtained. Most of the drug molecules will be having a log p value less than 5. So similar to P, aqueous solubility, otherwise what you call as log of S solubility, log of S, is going to be a very significant aspect which we measure in the molecules. And if the molecule is going to be having a low solubility, what does it mean? It may not be absorbed properly, right? So that means that absorption of a drug molecule, that depends upon solubility, aqueous solubility. What I mean is aqueous solubility to a large extent. And it is found that 80% of drug molecules, or more than 80% of drug molecules in the market, non-drug molecules, that would be having a log S value greater than -4. So with this itself, we got some idea. Molecular mass, it should be less than 450. It should not be more than 450 at least. Log P value, we put it as it should be less than 5. Log S value, it should be higher than -4. So next comes polar surface area, PSA, and it's a very important measure as far as drug likeness is concerned. I've seen that if the polar surface area is going to be more than 140 square angstrom unit, it would be a poor uh, uh, drug molecule. Especially, it would be negligibly permeating through cell membrane. So absorption will be negligibly small. Bioavailability will be negligibly small. So automatically, the polar surface area should not be higher than 140. Is a condition for most of the drug molecule. At the same time, if you take any CNN drug molecule, central, the neural networking, then that means for the molecules to cross blood-brain barrier, you see that polar surface area should be less than 60 square angstrom. So we can set up two conditions. If the polar surface area is going to be less than 60 square angstrom unit, it would be able to pass through blood-brain barrier. If the polar surface area is going to be less than 140, it can be considered as a probable drug molecule. It should not be anyway greater than 140, isn't it so? If it is going to be higher than 140, that permeability would be negligibly small. Now these are some of the features like hydrogen bonding donor, hydrogen bonding acceptor, then molecular mass, log P value, log S value, polar surface area, etc. They are the common primary features which we normally take while making the screening of molecules. At the same time, so as to get some lead molecules, or so as to identify some lead molecules, some rigorous screening technique is required. And in this connection, 3 normalized activity measures, what you call as bioactivity measures. So we call them together as bioactivity measures. They have been introduced, and actually through these 3 properties, to a large extent, we will be able to explain the behavior. So that is the idea behind introducing bioactivity, criteria or features. And they are actually, first one is what is known as ligand efficiency. Second one is lipophilic ligand efficiency. So ligand efficiency is normally represented as LE. lipophilic ligand efficiency LLE, and finally there is a measure what you call as ligand efficiency lipophilic price LELP, ligand efficiency lipophilic price. Now let us see them one by one. Ligand efficiency is actually is a free energy in kilocalorie per mole per non-hydrogen atom. It is normally calculated from the experimental value or IC50 value, and of course the ligand efficiency, as that free energy increases, we can say ligand efficiency increases is a measure of activity that is normalized by the number of non-hydrogen atoms. Okay, and the unit will be kilocalorie per mole per non-hydrogen atom. It's a very primary factor, and the drug, while interacting with protein, we'll be getting drug protein complex. So chemically that process is a reversible process. It can be represented as drug plus protein gives drug protein complex. That forward reaction, that speed is sometimes referred to as small letter k on, and backward rate constant is known as k off. K on is actually the rate constant determining the formation of ligand complex or ligand target complex. Ligand target complex. Okay. Off means actually is a rate determining the dissociation of that complex. So formation of that complex is represented by rate constant k on, and dissociation of that complex is determined by rate constant off, k off. Equilibrium constant for that process, that is actually active mass of that complex, drug protein or drug ligand complex, drug protein or drug target complex upon the active mass, product of active masses of ligand and target, ligand and protein or ligand and target. Okay, now normally activity or affinity of a drug molecule, affinity of a drug molecule towards its biological target, it is normally measured not through this equilibrium constant. Actually, this equilibrium constant is normally known as equilibrium constant binding, so Kb. At the same time, inverse of Kb, 1 by Kb, is actually its dissociation constant. Theoretically, it is dissociation constant, right? And this dissociation constant is actually a useful measure of computing affinity of drug molecule, or what you call as efficiency of drug molecule. Actually, the number, actually the Kd, as the Kd increases, what would be the result? As Kd decreases, what would be the? So very easily we will be able to interpret. So actually speaking, what we can say is As the KD increases, KD, what would be that? It is the dissociation constant. So you may have to say the active mass of ligand into active mass of target upon active mass of complex. So as that increases, concentration of drug molecule in the dissociated form will be much more than concentration of drug molecule in the complex form, right? And we know that for the drug to act on the target, biological target, it should be associated with it, right? So automatically we can say drug action or the affinity of drug molecule towards biological activity is inversely proportional to dissociation constants. So normally, for all useful drug molecules, Kd value will be approximately 1 into 10 raised to minus 6. Otherwise, what do you call as? It should be in some micro values. Micromolar concentration should be there. And approximate range micromolar concentration should be there. In some cases, it may be nano level concentration, 10 raised to minus 9. Or in some cases, it will be even pico, 10 raised to minus 12. So we have to see that the required concentration of drug molecule in the body for a proper drug action is a very small quantity, 1 into 10 raised to minus 10, minus 6. Means it can be in the micro level concentration, micromolar level concentration, nanomolar level concentration, or picomolar level concentration. And what we have to see is, we have to see the dose that is required for providing this concentration in the body. Okay. So suppose you say that a 200 milligram drug dose. What does it mean actually? If 200 milligram drug is going to be given to the patient, then this would be absorbed, and after the absorption, this would give some micromolar concentration within the body, which would be sufficient for the drug action, right? Then the lipophilic ligand deficiency is the second property. So as far as ligand deficiency is concerned, which all points you have to remember? Ligand deficiency, then that depends upon the log P value basically. Then it depends upon absorption. Now let us take the lipophilic ligand efficiency. If the lipophilic, if the lipophilicity of a molecule is higher, we know that lipophilicity higher means log P will be higher. Its bioavailability will be comparatively less. Means along with say an increase in lipophilicity, there would be a decrease in bioavailability, right? So a gain in lipophilicity, it should be compensated with a loss in bioavailability. So these two things, they have to be taken together. Now you know that as the lipophilicity increases, bioavailability decreases. Suppose bioavailability decreases, how can you increase affinity? Affinity can be increased if and only if activity of the drug molecule is more. So a compensation could be made only by increasing activity of the target. Target activity should be increased. Or what you can say is bioactivity should be increased. Not the target activity, bioactivity of that drug molecule within the target or towards the target, it should be increased. Right? So actually, just to understand it fully, this lipophilic ligand deficiency term has been introduced. It's a very simple expression, that is minus log of IC50 minus C log P. C log P is actually log P, measure of log P, measure of lipophilicity, and IC50 is actually a measure of the bioavailability as well as activity. So we introduce a new term, what you call as lipophilic ligand efficiency. Lipophilic ligand efficiency should not be highly lipophilic. For the efficiency of drug molecule, it should not be highly lipophilic. So that is why a new term has been introduced, what you call as ligand lipophilic efficiency. And actually the a rough rule of thumb suggested by Jonathan E.S. Mason et al., which appeared in Nature Biotechnology, is, see, if the lipophilic ligand efficiency value is above 3, it will be a lead molecule. Suppose it is going to be more than 5, it's going to be a clinical candidate. So in most cases, directly we will be able to use that molecule. So with that, actually lipophilic ligand efficiency is going to be a very important term for us, because if you measure lipophilic ligand efficiency, then any lead molecule will be having LLE, lipophilic ligand efficiency, more than 3. Suppose any other molecule, any molecule is there identified with less than 3 value for LLE, immediately you can neglect them. It would not be a lead fit molecule, fit to a lead molecule. Similarly, suppose it is going to be more than 5, you can make that sure that this is going to be a clinical candidate. At the same time, we have to see that with these two terms, whether you are completely satisfied or not, not at all again. Because when you consider the ligand efficiency, we know that ligand efficiency doesn't include lipophilicity into account, because it's some energy, it's a thermodynamic term basically, free energy associated with breaking down of non-hydrogen atoms. So it's actually neglecting lipophilicity into it, whereas in the lipophilic ligand efficiency, that term is included. Still, the problem is it neglects molecular weight influence. See, ligand efficiency is not suitable for, or considering it as a screening feature, because it doesn't take lipophilicity into account. Lipophilicity is considered in the lipophilic ligand efficiency. At the same time, here molecular mass is not going to be considered. Hence, Kesaru, Makara et al., they suggested a new feature, what do you call as LELP. LELP is actually ligand efficiency lipophilic price. LELP, ligand efficiency lipophilic price. Now let us see that actually the term is very simple. Ligand efficiency lipophilic price is log P divided by ligand efficiency. It's a very useful function which will tell us the price of ligand efficiency paid in log P. Normally, you know that from the definition itself, you know that if the log P value is going to be negative, then LELP, ligand deficiency, that is going to be negative, right? At the same time, in the ligand efficiency lipophilic price, it can be positive. See, negative value, only one possibility is there. Why? Reason is, you know that the term is defined as log P divided by ligand efficiency. Ligand efficiency lipophilic price. Log P divided by. So if log P is going to be negative, definitely we can say LELP maybe LELP will be negative, right? And in that case, we may have to conclude like, okay, higher the absolute value of LELP, that molecule would become less, or we can say lesser drug-like that molecule would be. Okay. So the accepted lower limit of ligand efficiency, it is less than, it is 0.3. It is a lower accepted limit of ligand efficiency. So with that, if you measure, what we can say is, as a condition for lead like molecules or lead like compounds is log P value should be between -3 and +3. Okay. And if you substitute these two things in the definition of LELP, because ligand efficiency lower limit, that is 0.3, and I told you lipophilicity range is -3 to +3, so automatically LELP range will be -10 to +10, right? So we can say that the range for LELP, ligand efficiency lipophilic price, should be between -10 and +10. If it is going to be minus, if suppose the LELP is going to be minus, log P value will be negative. That also you have to remember. And mind you, for the Lipinski zone, Lipinski is a very primary screening criteria. If you want to include the entire molecules listed in the through Lipinski's rule, what do you call as Lipinski zone? If you measure LELP, it would be up to 16.5. It needs not be up to 10. But based upon the later classification and data mining results, we got it as up to 10. So in most of the lead discovery projects, what we see is closer LELP is to 0 in the positive range, the better. So based upon that, what we can say is for most of the compounds, ligand efficiency range is between 0.4 and 0.45. Log P value is between 0 and 3. LELP value is between 0 and 7.5 for most of the compounds, if you see. But mind you, it should not be negative. If it is going to be negative, log P value is negative. If log P is negative, problem is that molecule is going to be highly ionizable. Isn't it so? If the compound is ionized, then there is every chance for that molecule to be deposited within the cell membrane, because you know that when the drug molecule passes through the cell membrane, it's actually a bipolar membrane, isn't it so? Aqueous layer is there, then organic layer inside and outside. At the center, aqueous layer is there. So drug molecule passes through, it gets into, once it reaches the aqueous level, the problem is it may be precipitated, because you know that when the ionic product becomes higher than solubility product, that molecule will be precipitated. So this results in, actually, if it is going to be negative, major problem would be there is a chance for that drug molecule to be precipitated within the cell membrane, that blocks passage of any material through cell membrane. So that is why, though from -10 to +10 they are all lead molecules, if you carry forward the concepts, we may have to conclude that it should not be negative. And even up to 10, sometimes you will not be able to take, because for most of the molecules, up to about 7 to 7.5 only came LELP. So that the most suitable screening strategy is make LELP value between 0 and 7.5. So actually, a typical lead discovery project means it's actually a move towards 0 LELP value, from higher value to 0. If it is 16.5, that will be in the Lipinski zone. Put it down again, make it little less. I told you, suppose it is going to be 10. Again, we say that most of the drug molecules would be having, most of the personally available drug molecules would be having LELP value less than 10. Okay. Again, what you see is bring it again down, and most of the drug molecules, we may have to say more than 80% of drug molecule would be having LELP value less than 0, less than 7.5. So that is why what we say is, so in the broader sense, what we can say is the drug discovery project as such is a move towards bringing down LELP value. So I think these three components also would be added into your project: ligand deficiency, lipophilic ligand deficiency, and LELP. And we will make a very strong screening to get the most suitable drug, most suitable lead molecules. And that part I shall demonstrate through the data barrier. Okay, so we will take the parent database which we made in the last class. So let me take open, recent, then euphrasis officialis, phytochemicals, new database, and that data has been uploaded here. And you may remember that data. So what we have is, we have only few information here. Structure is there, name, then molecular identifier, then total molecular weight. These are the only properties kept here for the entire molecules which we identified. So 22 molecules we identified in that medicinal plant. Now, first thing is, let us generate the properties from chemical structure discretase. Then we can generate properties. Okay, properties. Drug likeness related molecular weight is already there. So let us have C-log P, C-log S, hydrogen bonding donor acceptor, total surface area, relative polar surface area, the percentage will be given to you. Then drug likeness sufficient. Ligand efficiency wise, we have the ligand efficiency, then lipophilic ligand efficiency, and ligand efficiency lipophilic price. Okay, other things we can keep as such. If you want, you can generate non-hydrogen bonds, number of non-hydrogen atom count, etc. But we want the drug likeness to be named through these descriptors: C-log P, C-log S, hydrogen bonding acceptor, hydrogen bonding donor, total surface area, polar surface area, drug likeness. Mainly these three properties we want to add: ligand efficiency, lipophilic ligand efficiency, and ligand efficiency lipophilic price, right? So let us generate them. In few seconds, these properties will be generated and properly kept here. So you are seeing at present C log P, C log S, hydrogen bonding acceptor, hydrogen bonding donor, relative polar surface area, drug likeness, ligand efficiency, lipophilic ligand efficiency, and lipophilic ligand efficiency lipophilic price, otherwise what you call us. Okay, now if you go through this, see, we can use some primary feature like hydrogen bonding donor, hydrogen bonding acceptor, etc., and make a primary screening like hydrogen bonding donor. Our requirement is up to about 5 we can have, right? Here it is up to some 10 or 11 or 16, up to 16 is there, 46 is there, 191 is there. It's a very big molecule. So automatically such molecules you can immediately remove. Hydrogen bonded donor wise also, what you see is up to about 116, and you know that hydrogen bonding acceptor normally it will be less than 10, donors will be less than 5. Uh, Polar surface area, relative polar surface area is given to you about 0.38 percentage. So automatically if you put it as 0.4, about 40, that would come about 40 polar surface area. So like that you can count in all these cases, or if you want you can just multiply these two and get another one here. Drug likeness, what you do is negative drug likeness you will remove, but here the problem is you have negative drug likeness for a number of molecules. Listen. And I told you about LELP, ligand deficiency lipophilic race. The expected range is actually from 0 to 7.5 or 0 to 10. And we can even think like, okay, it can be something like up to 15, you can think. That's 0 also, there also. If it is close to 0, then also you may be able to add that value. Because the only problem is LELP negative means actually that log p value will be negative, right? But if it is going to be very close to 0, that also we can think. And especially the primary screening. Still here what we see is few molecules are there. This is positive, positive. Here also positive. It's slightly higher than 10. All right.
That molecule seems to be a probable molecule. But if you take drug likeness slightly negative, right, minus 0.08. So such things you can make some relaxation because rules are not very rigid, and few molecules you can very easily identify from here. And the most suitable molecule you may pick up, and even that inter relationship that can be generated either by using a two-dimensional diagram or a three-dimensional diagram. So let us initially make a two-dimensional presentation where structure will be given along the x-axis. Then y-axis, let us give LELP. Okay. Now what we do is we will give some colors. So set marker color. Then for LELP, let us take it as a standard marker, LELP, and color by category also. So we'll be able to see the different categories here. So let me get the full screen. So here what I see is actually these are the different categories which I identified in this 22, and I can very easily I can see the values here. Here it is mentioned. Similarly here also for the color code we'll be able to see them. And if you want to see the relationship between any two properties, similar to structure, if I want some other property like C log P, that correlation we'll be able to see. And what you see is most of the molecules have got C log P value within some range close to 0. I mean, few of them slightly higher than 0. Then suppose hydrogen bonding, there also what you see is it is the positive level. That means that intercorrelation we'll be able to set up very easily. So similar to that, if you pick up a 3D picture, so three variables could be put up together: structure, then second one, let us say LELP, and the third one is, let us take drug likeness. Here also we can generate the color cards with respect to LELP. Different colors are there. So this is actually the ligand sphere. Here what you see is one molecule is outside this, which has got a very low LELP value. And if I want to name these molecules, that is also possible. What I can do is, instead of structure, I can put the dataset name. So I'll be able to get the name, and so instead of name, suppose I need only that molecular name, so that the number will be there. That particular number will be mentioned. Approximate. So whatever derivative or whatever component is required there, here you see that one molecule has got a very unexceptional, or what do you call as violating a LELP rule, and that molecular name is going to be 24763. 24763. We keep it here. 24763, this molecule, aniline. So there is no chance for aniline to be taken as a drug molecule. Similarly, I have few molecules which are there in the ligand sphere, or what you call as the drug sphere, drug space. You may call it as a drug space. So this is actually the drug space, and the major thing which you have to remember is here also you are seeing one molecule here. Okay, the major thing which you have to remember is there is no rigid rule as far as the screening are concerned, but definitely it will give you some idea about the relationship here itself. Suppose you want to see the relationship between LELP. Suppose I take instead of drug likeness, if I take hydrogen bonding acceptor. So what I see is the molecules, their dependence on any molecules dependence on hydrogen bonding acceptor, I'll be able to see. So instead of hydrogen bonding acceptor, if I take hydrogen bonding donor, almost the same relationship is going to be there. Then suppose I take a C log P, so I can see that the molecule accepting one or two molecules, it will be there in another space. So the correlation between LELP and molecular structure with respect to different parameters I'll be able to generate. And so in this manner, the medicinal plant which you have identified, there also you may identify the most suitable molecule, generate these attributes and identify the most suitable molecules, then make the evolutionary molecular database. There also you follow same procedure and come up with few highly suitable molecules and report to me. Okay, thank you very much.